I'm just cutting this DNA molecule with the help of a molecular scissor because I want to reconstruct a genetically modified Spider-Man or Spidey. If not Spider-Man, obviously we can construct genetically modified mice, genetically modified tomato and other genetically modified organisms. In this chapter, we'll also learn the process of spooling of DNA from a cellular fraction. Yes, I'm talking about the class 12 chapter Biotechnology Principle and Processes. I am Bishwajit, your zoology mentor, and we are in PW NEET English. Here we learn with fun. And after watching the complete video, we need to solve or practice some questions like statement based questions as well as assertion and reason type question. And the source is PW NEET app. Just click the link given in the description box. So without delaying time, let's reconstruct DNA, let's manipulate DNA and learn how to form recombinant DNA by studying recombinant DNA technology. So the topics we're going to cover in this chapter, we'll start the chapter with introduction, that is definition of biotechnology. We'll try to differentiate between old and modern biotechnology. We'll talk about the principles of biotechnology. There are two important principles we need to talk about. We'll learn the tools to form recombinant DNA or rDNA, the process of formation of recombinant DNA, and we'll conclude the chapter with the topic of bioreactors. These are special facilities in laboratories where we use genetically modified organisms to produce products like enzyme, antibodies, antibiotics and other proteins. Okay. So before starting this unit of biotechnology, there are two chapters under this unit. We need to learn about some introductory talk. So at the introduction of this unit, you will get the name of this particular scientist, René Descartes. Yes. So basically, he was a scientist from 17th century and he was a French philosopher come mathematician come biologist. So what is the contribution of this gentleman? Basically, he tried to form a bridge between the natural science and the application based science, right? So all human knowledge, especially the natural sciences. Now what do you mean by natural sciences? It's mathematics, physics, chemistry and biology, were directed to develop technologies which add to the creature comfort of human lives as also a value addition to our life. So he is talking about, here we are talking about how the study of nature helped us to redefine our culture, redefine our advancement, redefine our scientific knowledge towards the lab to land based study, towards the application based study. Okay, so here we can see the whole approach to understanding the natural phenomena become anthropocentric. Now what do you mean by this, the word anthropocentric? So basically René Descartes, he talked about application. Now the application of the knowledge whatever we have learned by studying those biology, physics, chemistry or mathematics. Okay, so for the first time when human observed a bird is flying, so they tried to learn the scientific reason or the physics behind that and they invented aeroplane. You already know about Wright brothers. Okay, so that is the anthropocentric concept of our learning or learning outcomes. Yes, so when you're talking about physics and chemistry, they gave rise to engineering, technologies and industries which all work for human comfort and welfare. Okay, So if we talk about our comfort in our daily life, whatever gadgets or goods you are utilizing, you are watching this video in your mobile screen or laptop screen or your computer screen, you are sitting inside a air conditioned room with AC, electronic goods like fans, and you are reading under the artificial light of tube lights or bulbs. So everything is boon of science, right? Now, what is the major utility of biological world? The answer is here, as a source of food. The major utility of biological world is as a source of food. We have studied the nature. We have studied different organisms. We have studied microorganism, and then we have learned multiple microbe mediated process which help us to produce numerous items in our food science, right? Biotechnology, the 20th century offshoot. So biotechnology is 20th century offshoot of modern biology, changed our daily life as its products brought qualitative improvement in health and food production. So there are two ways. One is quantitative way and another is qualitative way. Quantity means drastic improvement in the production of food in the terms of blue revolution, in the terms of green revolution, in the terms of white revolution. These are the major success of classical biology, right? But now we have to choose, as because the population is outbursting, and now our need is, along with the quantity, we need quality. We need good proteins, good carbs, good lipids in our plate, right? So we are searching for quality over quantity. Now we need the help, or we have to grab the hand of biotechnology. Okay, so whatever we have learned from this previous slide, we'll try to figure out or try to frame some questions from that. Yes, as per the new trend, questions can be asked from anywhere and everywhere inside our NCERT. So basically it's the assertion and reason based question. So the assertion says that the whole approach to understand the natural phenomena became anthropocentric. Absolutely true fact. Reason is said all human knowledge, especially the natural sciences, were directed to develop technologies which add to the creature comfort of human lives as also value to human. This is also true, and we can say that the reason is justifying the assertion. Yes. Now another statement based question. Now the statement 1 says that the major utility of biological world is as a source of food. Exactly, it's a true statement. The major utility of biological world is as a source of food, but nowadays we are also developing towards our human welfare and health science, right? Statement 2 is saying that biotechnology, the 17th century offshoot of modern biology. Is it? Biotechnology is not 17th century offshoot. Biotechnology is basically 20th century offshoot. So this is a wrong statement. So this will be false. Okay. Now next, we'll talk about these two prominent pioneer scientists of biotechnology. Though the father of biotechnology is Carl Ehrlich, his name is not mentioned in our NCERT, but obviously these two scientists, one is Herbert Boyer and another is Stanley Cohen. Okay, now why we need to study about them? We are talking about recombinant DNA technology. We'll talk about genetic engineering. So it was the first case, for the first time in this world, Herbert Boyer and Stanley Cohen, they had constructed a recombinant DNA by combining multiple genes or multiple pieces of DNA and successfully transformed a host cell, right? So that's why we need to talk about them. So basically when I'm talking about Herbert Boyer, he was an expert of restriction endonuclease. He was an expert of REN. REN means restriction endonuclease. These are molecular scissors used to cut the piece of DNAs. Okay. Now, what about Stanley Quinn? He was an expert of plasmids. Plasmids we will talk about. They are biological, they are vectors or gene taxis or vehicles we use to transfer gene from one place to another place. So basically when they met with each other, their knowledge sharing had given rise to a new stream of biology, right? That is rDNA technology, recombinant DNA technology. So by 1969, I'm talking about Herbert Boyer. He performed studies on couple of restriction enzymes. Basically, his favorite one was EcoR1. His favorite restriction enzyme of E. coli bacterium, which especially useful properties. So Boyer observed that these enzymes have the capability of cutting DNA strand in a particular fashion, which left what has become known as sticky ends or overhanging stretches on the strands. Now these clipped ends made pasting together pieces of a DNA precise exercise. So basically, if I am talking about a piece of DNA, so DNAs are obviously DNA molecules. They are double-stranded structure. So with the help of REN, restriction endonuclease, we can cut these DNA molecules in specific sites, right? So that's why we call these molecular scissors, and they form a special kind of cut that we are calling the sticky end cut. So these overhanging stretches, these overhanging stretches are called sticky end cuts. So with an example, I am trying to show it. So suppose this is a complete DNA. Now after cutting them or cleaving them with the help of restriction endonuclease, we'll get sticky end cuts. You can observe these are the sticky ends. Now with the help of ligase, a pasting enzyme, you can attach them very easy manner. So before the study of this particular sticky end cut of restriction endonuclease, pasting to different piece of DNA, it was a hideous task. So that's why we need to talk about Herbert Boyer, his pioneering work on cutting and pasting DNA molecules, right, or DNA strands. Now we'll talk about Stanley Quinn. So when I'm talking about Stanley Quinn, Quinn had been studying small ringlets of DNA called plasmid, and which float about freely in the cytoplasm of certain bacterial cells and replicate independently, remember, form the coding strand of DNA. So Quinn had developed a method of removing this plasmid from the cell and then reinserting them in the other cell. So if this is a bacterial cell and this is the normal genome, now sometime we'll get these ringlets autonomously replicating double-stranded extra chromosomal DNA. So these ringlets are called basically plasmids. Okay. Now what Stanley Cohen did? Successfully he took out this plasmid from suppose this is a species of bacteria named A, and he successfully inserted this to another species of bacteria. Suppose this is named as bacteria B. We successfully reinserted the extracted piece of ringlet or plasmid into the bacteria B. So that was the success of Stanley Cohen. So we will say now this bacteria is transformed. From where this bacteria got this piece of ringlet? From bacteria A. So that was the success of Stanley Cohen. Okay. So now if you combine these two studies of different scientists, Boyer and Cohen, that will help us to reconstruct a DNA molecule, and gradually we will learn how they did this. Okay. Now we'll check our learning outcome by solving some questions. Okay. Another assertion and reason kind of question is given here. Okay. So the assertion is saying, before Boyer, pasting pieces of DNA together was not a precise exercise. It was a hideous job, right? So assertion is absolutely true. Now the reason is saying, because Boyer observed that a couple of restriction enzymes have the capability of cutting DNA strands in a particular fashion known as sticky ends. This is also true, and yes, the reason is correct explanation of our assertion. Another assertion and reason, let's check it out. So the assertion is saying Cohen had developed a method of removing plasmid from the cell and then reinserting them into other cell. Absolutely true. The reason is saying that combining this process with that of restriction enzymes enabled by Boyer, recombinant DNA was produced. So if we try to link these two statements, both are true. So Cohen had developed a method of removing plasmid and reinserting them into other cells, because combining this process which that of restriction enzyme, or this reason is saying about the process which was supported by Boyer. So reason is true, but reason is not describing or elaborating the assertion. So we can say about this question, assertion and reason both are true, but reason is not the correct explanation of assertion. Okay. Now let's jump into the chapter itself. So what is biotechnology? Probably we are talking about old biotechnology. Yes. So biotechnology deals with techniques of using live organisms or enzymes from organisms. So biotechnology is the technologies of using live organisms or enzymes from organisms, like if you can take examples of microbes, to produce products, and processes which are useful to humans. Yes, like for one example I am giving, to make curd from milk we use lactobacillus bacteria. If not lactobacillus bacteria, we can extract the lactic acid produced by them and we can convert milk into curd. So basically when you are talking about examples, they are basically microbe mediated process, microbe mediated processes, and like formation of curd, wine and bread, and also you can incorporate the formation of idli, dosa, the industry of fermentation technology, right, by using fermentation. When you are talking about fermentation, we need the help of microorganism. So they falls under old biotechnology. Here this is your favorite thing of many of you, right? Curd, and obviously the breakfast we are doing most of the days, that is with bread. That is, these all the things, these are boon of biotechnology or fermentation technology. But we have to upgrade our knowledge, we need advancement. So while we are talking about today, biotechnology is in more restricted sense. In more restricted sense, biotechnology means in vitro fertilization. This is the methodology to form a test tube baby, right? They will fall under IVF technology. In detail we have learned in the chapter of human reproduction and reproductive health. Okay, synthesize and using of a gene, like we can talk about, for one examples, by utilizing the insulin forming gene of human being, we can convert bacteria into genetically modified bacteria, and we can use those bacteria as the host to produce human insulin, right? Preparation of DNA vaccine, recombinant DNA vaccine, they are the second generation vaccine. Directly inserting DNA or RNA, they are the third generation vaccine. So second gen and third generation vaccine, we need help of recombinant DNA technology or biotechnology. While we are talking about correcting a defective gene, that is called gene therapy. When you're talking about gene therapy, we need again help of rDNA technology or biotechnology. So these are some products of modern day or today biotechnology. DNA vaccine, one of the vaccine which was prepared for COVID-19, right? Formation of embryo in lab with the help of biotechnology. You can talk about in vitro fertilization, you can talk about ICSI, intracytoplasmic sperm injection technique, the technique of gene therapy in embryonic stage. Few congenital disorder can be eliminated with the help of gene therapy, and formation of other genetically modified organisms like genetically modified corn, genetically modified cotton, genetically modified brinjal, genetically modified potato, all are the boon of modern biotechnology. So here again we will check our knowledge with the help of one question. So find out the odd one. So the question is asking, in a restricted sense, biotechnology means, so in a restricted sense means here they are asking about modern biotechnology. Is it use of genetically modified organism? Yes. Is it in vitro fertilization? Yes. It's a part of modern biotechnology. Is it microbe mediated process like making wine and curd? No. Making wine and curd, these basically this process comes under old biotechnology. Long back we already discovered the process to form curd, to form bread or wine, right? Now what about DNA vaccine preparation? That is also comes under modern biotechnology. So our answer will be, the odd one will be option 3. Microbe mediated process are not considered as a part of modern biotechnology but old biotechnology. Okay. Now, to fill the gap between the old biotechnology or the natural science or traditional one and the modern one, we got name of one organization called European Federation of Biotechnology. European Federation of Biotechnology. Okay. So what they tried? They tried to merge traditional view with modern biotechnology. So we need to study their definition of biotechnology by EFB, the integration of natural science and organisms, cells and parts thereof, means cell organelles and molecular analogues. Now what do you mean by molecular analog of cell? Here they are talking about biomolecules for products and service. So basically that is a punch of old biotechnology and modern biotechnology. Obviously when you are talking about biotechnology, we need to start it with cytology or cell biology, right? We need to modify the genes, DNA or RNA, the chemistry of gene inside a cell, right? Okay. So the topic we are going to talk, that is principles of biotechnology. So there are two core principles. There are two core techniques or two core principles we have to follow when you're talking about biotechnology. The first, that is called genetic engineering. And the second, that is bioprocess engineering. Or you can call it biochemical engineering. Genetic engineering and bioprocess engineering. So what do you mean by genetic engineering? Basically, this is the manipulation of genetic material. Means also can be called as alteration of chemistry of DNA and RNA. This is the first thing you have to do in genetic engineering. Alteration the gene or the sequence of a gene or DNA, then inserting that modified gene, that is called rDNA, recombinant DNA, into a host organism. Obviously, what is host organism you are questioning? Host means the organism which we are gonna culture in our lab by utilizing the bioreactors to obtain our products, right? And when you will incorporate the modified gene into the host organism, that will bring a change into genotype, and ultimately that will change the phenotypic expression of that host. So, next thing, that will be changing phenotype expression in the host organism. So, as I was saying that we can take out the insulin producing gene from human being and incorporate with the genome or with the chromosome of E. coli bacteria. Now, if you culture that genetically modified E. coli bacteria that is acting as a host in a bioreactor, so that will produce human insulin for us. Okay. Now, what is bioprocess engineering? So, in genetic engineering, we produce the product from the genetically modified organism in a very low scale, in small scale. Now, if you really want it to produce in a large scale, in economical scale, or for the human welfare, so you need to produce them in a large quantity. So, for that you will be needing a facility, special facility with sterile laboratories. So, bioprocess engineering, that means maintenance of sterile ambience What do you mean by sterile ambience? Sterile ambience means microbe contamination free environment into your laboratory, because you just wanna culture the target host organism only inside your lab. You don't want the growth of unwanted microbe. Like if when in a large scale, suppose in another example I am trying to make it easier, in a agriculture field you are cropping for wheat plant, right? So you want, what is your target crop, target plant, that is wheat plant. But along with wheat you will find that many other herbs are also growing along with wheat. They are disturbing your agriculture, right? So you will try some methods to eradicate. You will spray some herbicides to eradicate those weeds from your agricultural field. So similarly, when we are culturing microbe into our lab, some unwanted microbe will create hindrance or will create annoyance into our lab culture. So for that we need to follow the proper bioprocess engineering. So here we maintain sterile ambience for the growth of prokaryotes, means bacteria, or eukaryotic cells. Why you are culturing them? To obtain their product, like in the form of enzymes, in the form of hormones, in the form of antibiotics, in the form of other drugs, in the form of other proteins, etc. So remember these two principal technique of biotechnology. Now let's try our knowledge outcome. So the statement 1 is saying that genetic engineering deals with techniques to alter the genotype of the host organism. Yes, obviously when you are inserting a new gene, there will be change in gene pool, so there will be change in genotype as well as there will be change in phenotype. So that's a true statement. Now the statement 2 is saying genetic engineering deals with techniques to introduce gene of interest into host organism. Yes, the alien gene or the foreign gene is also called as GOI, gene of interest into the host organism. So this is also a true statement. Now another assertion and reason based questions, let's solve it. So assertions is saying that maintenance of sterile ambience in chemical engineering process is necessary step of bioprocess engineering. Absolutely true fact. The reason is trying to say that because it enables growth of only the desired microbe or eukaryotic cell. Yes, that is the important word. We want the growth of only our target organism, that is desired microbe or eukaryotic cell, in large quantities for the manufacturing of biotechnological products. So that is also absolutely true, and the reason, yes, the reason is satisfying or explaining the assertion. Okay. Now, what is the advantage of sexual reproduction over asexual mode? So basically, all the varieties we are creating, the variation in this ecosystem or artificially, they can be artificial variation, they can be natural variation, they are boon of sexual reproduction. So what is the difference between sexual reproduction and asexual reproduction? So when I am talking about sexual reproduction, that basically promotes or permits variation. Sexual reproduction permits variation. Okay, how it permits? It's permitting variation or promoting variation. During the time of sexual reproduction, a male partner will produce male gamete, female partner will produce female gamete, and during the formation of these gametes that we call gametogenesis, genetic recombination takes place. Genetic recombination takes place, and due to this genetic recombination, there will be appearance of some new characteristics, features, or new expression of some new traits into the offspring. Okay, so that is variation. So one of the very easiest example I am giving you. Suppose your mother is very fair in color, your father is dark in color, but you are neither so much fair nor so much dark. So you have a mediocre complexion in your skin color. So that's so you will be saying that you are a variety, new variety. So suppose you have some pet rabbits. One rabbit is white in color and another rabbit is black in color. But when they are reproducing their offspring, they have a grayish color. So that's because of the genetic recombination. Now, why this recombination takes place? So during the time of gametogenesis, okay, the meiosis 1, or during the time of first meiotic division of this gametogenesis, there will be exchange of DNA in between homologous chromosomes. Yes, you know that we are a diploid organism. We are diploid organism, but our gametes will be haploid. But during the formation of gamete, there will be exchange of part in between the chromatids of homologous chromosomes. Basically, there will be exchange of genetic material in between the non-sister chromatids. These are two homologous chromosomes, means one chromosome is from your father, another chromosome is from your mother, and they have similar type of gene present into them, right? So that's why you are calling them homologous chromosome. So during the time of crossover, there will be crossing over in between these two homologous chromosome. So after crossing over, what we will find that the arm has been exchanged. So the arm exchange will be there during the time of crossing over. During the time of what? During the time of crossing over in between the homologous chromosome, and that gradually creates the genetic recombination in the offspring. So basically the gene of father and mother get mixed up. So that's why we say that sexual reproduction that promotes variation and that creates the nave of natural selection and evolution. Now, when we are talking about a sexual reproduction that conserves genetic information, means if this is the parental generation, if this is the parental generation, the offsprings will be identical to it. The offsprings will be just exactly identical to the parent. There will be no genetic change into the offspring. So suppose this is the parent and these are the offsprings. So these offsprings are just identical with the genetic makeup of the parent, and they're 100% identical with their parents. So that's why there is no chance of variation or very less chance of variation. That's why we are telling that a sexual mode of reproduction conserves genetic information, while sexual reproduction permits variation, and the variation is the nave of natural selection and ultimately formation of new trait, and it will boost up the process of even evolution or formation of new species. Okay, so we'll choose sexual reproduction over asexual reproduction. Now let's solve some question. Again, assertion and reason based question is given here. Assertion is saying that sexual reproduction provides opportunities for variation and formulation of unique combination of genetic setup, and some of which may beneficial to the organism as well as the population. Yes, remember, some of the variations will be beneficial for the population or the organism, not all the variations. Okay, so this is obviously true statement. The reason is saying recombination promotes natural selection. Yes, as because this variation is accumulating generation by generation, it will support the process of evolution as well as natural selection. So this is also true statement, and yes, reason is justifying, reason is a proper justification for assertion. Okay, now another question, which of the following supports anthropogenic selection well as defined by Darwin? Okay, so naturally occurring, so here I am trying to connect this chapter with the chapter of evolution. So naturally occurring variation due to sexual reproduction, are the example of anthropogenic selection? No. Anthropogenic selection means we have done something, and as a consequence there will be formation of variants into a population. Okay. Traditional hybridization procedures used in plant and animal breeding techniques preserve the genetic information, they are talking about asexual reproduction, or recombinant DNA technology. Okay, so which of the following supports the anthropogenic selection defined by Darwin? So the answer will be the traditional hybridization procedure used in plants and animal breeding, like by the process of selective breeding technique, we have produced from the wild dog to the normal dog or stray dog type, that is a hound. Okay, then we have produced German Shepherd, we have produced Bulldog, we have produced a Doberman, we have produced Rottweiler. From if you talk about plants, from the wild cabbage type, we have produced cauliflower, broccoli, kohlrabi. So all of these are examples of anthropogenic selection, and obviously the traditional hybridization procedure is also involved into that. Okay, one of the example of hybridization also I am trying to say here, like if you talk about hybridization, suppose there is a cow called Sahiwal cow, that is an indigenous cow from India, that will produce 7 to 8 liters of milk per day. But there is another variety of cow which is present in the USA, suppose that is called Jersey cow. Okay, and that can produce 30 to 35 liters of milk per day. So what scientists they did a hybridization between the Jersey bull and Sahiwal cow, and the offspring, the hybrid offspring is called Jersey Sahiwal. Now that can produce near about 20 to 25 liters of milk per day. So that is the result. Jersey Sahiwal is a hybrid of Jersey cow and Sahiwal cow. Now if you are crossbreeding them, you will get Jersey Sahiwal. Now what your aim was to increase the milk production in your indigenous cow. Okay, so the NCERT is saying that what is the advantage of genetic engineering over traditional hybridization procedure? Now why we are following a hybridization procedure? To obtain new traits by the process of hybridization, and by obtaining new trait you can increase productivity, and this kind of increase in productivity is called as quantitative improvement. Okay, now what happens during the time of hybridization? Basically, with an example I am elaborating it. Suppose this is a chromosome of your indigenous cow, and this is the chromosome of the Jersey cow. Now you want to crossbreed, you want them to be like crossbreed, you want hybridization. Suppose this chromosome, this particular organism has a good gene, and this particular organism also has another good gene. Okay. Now, if I am marking green tick, they are good genes. And if I am marking red tick, they are bad genes. Right. So, let's see, when we are hybridized them, the offspring got the good gene from one of their parent as well as bad gene from one of their parent. So, got one good gene as well as one bad gene. Now, what will happen? Suppose the good gene that increases the production and the bad gene that decreases the production. Now, for this particular parent, the production increasing gene was present. So, already the gene is inherited into the offspring. This is the gene of offspring of the hybrid one. But along with this good gene one, along with this good gene, a bad gene is also inherited. Bad gene is also inherited. So, that will decrease the productivity. That will decrease the productivity. Right. So, that was the reason I was telling that indigenous cow that can produce suppose 7 liter milk, but Jersey cow can produce 30 liter milk. Now, when you are crossbreeding them, you crossbreed between the indigenous cow and Jersey cow. And the crossbreed is Jersey Sahiwal cow. Sahiwal was the indigenous. Sahiwal was the indigenous. So, for the Jersey Sahiwal, the milk production, you were expecting it will be like 30 liter, it will be producing 30 liter milk like Jersey cow, but it's producing only 18 to 20 liter milk due to the inheritance of bad genes as well, right? So, that's why here we need to choose quality over quantity. During the time of normal hybridization, there will be inheritance of good genes as well as bad genes into the offspring, but we need to follow the qualitative method now, that is can't be done by following this hybridization technique. So, we will go for genetic engineering. So, what genetic engineering does? With the help of genetic engineering, suppose this is one of the organism, and you have observed that organism has a good gene into this. This organism has a good gene into this. So, directly what you will do, you will take out this, you will cut, you will separate the good gene from the entire genome of this donor organism, and this particular gene is called gene of interest or GOI, gene of interest. So, we will take this gene of interest selectively by the process of these cutting techniques which will be utilized in biotechnology or genetic engineering, and you will insert them to the cell of or the genome of another organism, you will recombine, suppose you will recombine this good gene with a vector or gene taxi, and you will insert this particular into another organism. So, in this process, you are transforming a cell in a very targeted or selected manner, that you are only transferring good genes from one organism to another organism, for X organism, from X organism to Y organism. So that is the victory of genetic engineering or biotechnology. It is the victory of genetic engineering or biotechnology over, there is the victory of genetic engineering or biotechnology over normal hybridization technique, right? So I think it's clear to you now. Again, I am repeating, hybridization technique is a traditional technique which is followed for the quantitative improvement of crop production, or increase the productivity of plants among animals or plants. But during the time of hybridization, we have observed that good genes are also getting transferred into the offspring, as well as bad genes are also getting transferred. But you need to promote only the transfer of good gene, and here hybridization technique will not allow you for this. So you need something artificial, and obviously for that genetic engineering is advantageous for us, right? Okay. So let's move forward. Now let's check our knowledge. We'll try to match the column A with column B. So permits variation, obviously sexual reproduction, this permits variation. Inclusion of undesirable genes. Yes, hybridization. When we are performing hybridization, there will be inclusion of good genes as well as, or desired genes as well as undesirable genes. Okay, isolation of only desired gene. Isolation of only desired gene, that is rDNA technology, recombinant DNA technology, that promotes isolation of only desired genes. Preserve genetic information. Preserve genetic information, that is asexual reproduction, that preserves genetic information. So why again and again I am repeating the same things? Because I want these terms, this concept to get imprinted in your mind for a long term. Okay, now let's cope with the next hurdle. The question is there, do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Now, you don't know the whole process of biotechnology. Just you have learned that you cannot follow the hybridization technique because you want quality over quantity. You want only desired genes to get transferred from the donor organism to the host organism. So what you did, this is the this is the donor organism, this is the host organism, this is the host organism, this is the donor, and this is the genetic setup of the donor organism, and this is the desired gene or our gene of interest. This is our GOI, gene of interest. So what you have done, you have taken out the gene of interest from the donor organism. You have taken out the gene of interest. You have taken out the gene of interest or GOI, and you have decided you will insert it into the host organism. You will insert this to the host organism, and as because you don't have knowledge, you thought after the insertion of this lone piece of DNA or gene of interest, that will start multiplying inside the host, and that will produce the desired product for you, and even while this host organism will replicate, will produce its progeny, it will transfer this gene of interest into the upcoming generation. But that won't gonna happen. So the answer is there. What is the fate of a piece of DNA which is somehow transferred into an alien organism without following the proper method? So the fate. So I am talking about the fate. That will not survive, or not be able to multiply or inherited. Okay, if you randomly you are transferring a piece of DNA into one unknown organism, that is called alien organism. First of all, the alien organism, the cell functioning mechanism will attack and destroy that piece of DNA, or if not, there is no chance that the piece of DNA will get activated, like the DNA will not get transcribed to form the mRNA. So what is the procedure? How cell produces a protein? Do you know? It's the DNA. By the process of transcription, the DNA produces mRNA. By the process of replication, DNA produces more DNA, and by the process of translation, mRNA produces proteins. So this is the normal central dogma of a cell, and this phenomena will not be observed with this gene of interest. So what are the things we need to follow here, right? What is the proper methodology to transfer the gene of interest into an alien organism or into the host organism? So there are many criteria out there. Either you have to incorporate that gene of interest within the genome or chromosome of host organism, or you need to incorporate that piece of DNA with such a piece of DNA which has the origin of replication site, right? So here you need DNA with origin of replication site, which is naturally absent in this gene of interest. The second thing, you need incorporation of GOI, gene of interest, directly into the genome or chromosome. So the genome or chromosome of any organism that has inbuilt origin site or origin of replication site. So either you need to incorporate that gene of interest with such a DNA which has the origin of replication, or directly you need a process which will directly tag that gene of interest inside the chromosome or genome of host. Then only it is possible. If you are following these two techniques, if you are following these two techniques, then it's possible that you can successfully, the gene of interest will replicate or multiply inside the host or that alien organism. The alien organism also can be called as host, and inherit to the next generation. Then we will say the whole process of performing our DNA technology is successful. So is it clear? Again I am repeating it. Suppose you were busy in your work, you were studying into your study room. So suddenly you saw that someone outsider is entering into your room and playing with your gadgets, right? So what you will do? You will scold that unknown person and you will eliminate them out from your room because you don't know who is he or she. But so the similar thing will happen with that gene of interest if you suddenly unknowingly insert into just any cell. The cell will destroy it, right? But in another example, you observe that your mother is coming with one or your father is coming with one unknown child and introducing you that, yes beta, this is the son of your that uncle who is staying in Vijayawada or who is staying in Hyderabad. Then you will try to recall, okay, I have an uncle who is staying over there, so he or she must be the son or daughter of that particular uncle. So you will not be scolding them because he is entering into your room via your father or mother. So similarly, the via thing, the medium you needed here, that is either a plasmid, either a piece of DNA or plasmid which already has the origin of replication, or directly if you incorporate the gene of interest into the chromosome or the genome of the host organism, then automatically that will follow the cellular pathway and it will produce the product and it will also be inherited to the next generation. So let's solve some question for our to check our learning outcomes, right? Okay, again another assertion and reason based question is given here. So the question is saying the most likely the alien piece of DNA would not be able to multiply itself in the progeny cell of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. Exactly what we were discussing, right? So obviously this is a true fact. Now let's see what reason says. So in a chromosome or other than a chromosomal DNA, if there is a specific DNA sequence called the origin of replication, which is responsible for the initiating the replication. You know, wherever inside a genome, wherever the replication will initiate, you need to form a replication bubble or replication fork, right? So from where the cell will decide to initiate the replication or to form the replication bubble, that is the ori site or origin of replication site. So reason is also true. So let's try to link them. So alien piece of DNA would not be multiply into that alien organism, but may multiply or inherited with because in the chromosome or other than a chromosomal DNA. Yes, obviously, because it doesn't have the origin of replication. So our answer will be yes, reason is correct and is the perfect explanation for assertion. Okay. Now, the most important thing, let us focus on the first instance of construction of an artificial recombinant DNA molecule. Okay. So here we'll talk about the experiment of Boyer and Cohn. So in the year of 1972, scientists named Boyer and Cohn, they performed experiment on Salmonella and E. coli. Salmonella is the source of the gene of interest and E. coli as the alien organism or the host organism where they want to transfer this recombinant DNA for successful production of their targeted product. Okay. So while we are talking about Boyer's and Cohn's experiment, we need to talk about some cells. So the donor cell will be Salmonella. So this is the host cell, Escherichia coli. That is the host cell. So the recombinant DNA they are gonna produce, they will insert inside E. coli, which doesn't have that gene. Now, as a source of gene of interest, they have chosen the bacteria called Salmonella typhi, typhoid causing bacteria that has a plasmid which is carrying a tetracycline resistant gene, that is the gene of interest, right? And they use some vehicles to transfer the gene of interest inside the E. coli, and the vehicle was a plasmid called PSC101, which they obtained from another strain of Salmonella that is called Salmonella Panama. So which is source of gene of interest? This is our gene of interest present in Salmonella typhi. So that Salmonella, when you're talking about Salmonella typhi, it has a plasmid called R plasmid, and this R plasmid has our gene OI, gene of interest, that is TET R gene. Our gene of interest is what? Our gene of interest is tetracycline resistance gene, which is TET R. Like if you apply tetracycline, that's a very important antibiotic that can kill the growth of Salmonella bacteria, right? The typhoid causing bacteria. But few Salmonella typhi, as because they have a special kind of plasmid called R plasmid, and that plasmid has this special gene called TETR, that is tetracycline resistant gene, that will produce a protein that will neutralize the effect of that antibiotic called tetracycline. So you cannot kill this Salmonella typhi with tetracycline antibiotic, right? So this was the gene of interest, TETR. Now you need to produce a recombinant DNA by using our gene of interest, that is tetracycline resistant gene. Now as a vehicle, they had chosen Salmonella Panama, another strain of Salmonella bacteria. They have taken their plasmid. So plasmids are basically, these are gene taxi or the vector. Now what they will do, the plasmid they use, that is named as PSC101 plasmid, P for plasmid, SC for Stanley Cohen, and the lab number was 101. So they used this particular plasmid PSC101 to incorporate the tetracycline resistant gene. Now if you talk about this PSC101, that has another antibiotic resistance gene, that is kanamycin resistant gene or ampicillin resistant gene. So naturally this PSC101 the plasmid that has ampicillin resistant gene, now they have incorporated tetracycline resistance gene as well. Now successfully they had constructed an rDNA, recombinant DNA. So now this recombinant PSC101 that will act as successful vehicle, now they will treat E. coli with calcium chloride solution, cold calcium chloride solution, and they will give heat shock so that the membrane will become permeable, the membrane proteins will become permeable, that can give entry to the recombinant DNA, and successfully they will transfer this recombinant DNA, the piece of DNA, inside the Escherichia coli bacteria. So this is the rDNA that is transferred into our host organism Escherichia coli. Now if you culture this bacteria into your lab, suppose these are the bacterial colonies, and if you want to check that either this E. coli, the colony of E. coli, they are transformed or not, you have to check by giving the antibiotics, that is tetracycline or ampicillin, and if they survive, then you can confer that these are transformed. So that was the experiment performed by Hubbard Boyer and Stanley Cohen, or Boyer and Cohen experiment, right? So again I am summarizing it in a quick manner. So what was their target? Their target was to incorporate tetracycline resistant gene inside the E. coli bacteria, right? So where they will get this tetracycline resistant gene? They will get this tetracycline resistant gene from R plasmid, and from where they can procure R plasmid? From Salmonella typhi bacteria. Now they will take out this R plasmid. This is R plasmid from Salmonella typhi. They will cut this piece of DNA with the help of restriction endonuclease. With the help of restriction endonuclease, they will cut and they will take out gene of interest, that is tetracycline tetracycline resistant gene. And what they will do, they will insert this tetracycline resistant gene with inside another plasmid called PSC101. Now this PSC101 plasmid, they procured this plasmid from another strain of Salmonella, that is Salmonella Panama, and that has this PSC101 plasmid. They took out the plasmid from Salmonella Panama. They cut the plasmid with the help of restriction endonuclease again, and with the help of ligase, they will paste this tetracycline resistant gene within the PSC101 plasmid. Now that has become a recombined one. That means our recombinant DNA, recombinant plasmid. Now you can easily use this plasmid to transfer the genes inside the E. coli. Now, when the E. coli will successfully accept or permit this recombinant DNA to enter inside the cell, when you will make the E. coli competent. Now, how to make this bacteria competent? By treating the cells of E. coli with cold calcium chloride, and by treating that cell with 40-degree temperature, and then placing them into ice. It is called heat shock treatment. So why you are treating them with all these things? So that the cell wall and cell membrane of E. coli bacteria will be compelled to intake the recombinant DNA inside the cytoplasm. Now, while you have successfully inserted the recombinant DNA inside the E. coli, then we can say that this cells or this cell is now transformed or transformant, right? Now you have to check either these are transformed or not. So you will apply antibiotics like tetracycline and ampicillin inside the culture media, inside the petri dish. So if they manage to survive, then you can conclude that these cells are successfully transformed. Okay. Now let's solve some question. After every topic we are solving question. So basically this is the method of teaching. I am trying to elaborate while you are studying each paragraph or each topic from NCERT, how to curate, how to frame questions from them. Okay. So another statement based question is given here. Statement 1 is saying the construction of the first recombinant DNA emerged from the possibility of linking a gene enclosing antibiotic resistance with a native plasmid from Salmonella. Yes, this is absolutely true fact. Now statement 2 is saying that from E. coli, after isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. Again read it out. From E. coli, after isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. Yes, obviously we had isolated plasmid with antibiotic resistant gene, but that was not found inside E. coli. So statement 1 is true, but statement 2 is false here. Okay. So what ever we have learned, we can conclude our learning with some three basic steps to construct genetically modified organism. So you can hence infer that there are three basic steps in genetically modified organism. The first step, that is identification of DNA with desirable genes. First you have to identify in which organism from which organism you can procure the gene of interest. The second thing, introduction of the identified DNA into the host by making it competent. And the third, and very important, your job is not done yet. The third step is maintenance of introduced DNA into the host and transfer of the DNA to its progeny. Successful multiplication, that is successful replication and inheritance. That is very much important because if you are going for economic production or aiming towards the human welfare in the large scale, for large scale production of the products from these genetically modified cells or microbes, you need to culture them inside your laboratory, inside the bioreactors. So you need the presence of the target gene or gene of interest into your cell, into your GMO, genetically modified organism. Then if it's not present, then how you can produce? With another example, I am trying to simplify it. So we had started the lecture with the example of formation or creating artificial genetically modified Spider-Man. Suppose you are forming Spider-Man into your lab and you want to send them as a soldier of Indian Army. So you got a demand from Indian Army, you need 10,000 Spider-Man. So for that you have built a facility to form genetically modified, to produce genetically modified Spider-Man, though it's listening kind of awkward, but for instance of examples, I'm just elaborating it. So for the genetic engineering method, you have prepared one Spider-Man by inserting the cobweb of spider web producing gene into a human being. But whenever the produced Spider-Man will reproduce and produce his or her own progeny, then their child, the son and the daughters, should have that spider web producing gene inside them. Then only you can produce more and more number of Spider-Man, and then you will send them to Indian Army. Okay. Now let's solve a question here, assertion and reason based question. Assertion is saying that in recombination DNA technology, human genes are often transferred into bacteria or prokaryotes or yeast or eukaryotes. Yes, that is possible. Human genes can be transferred, right, in prokaryotes as well as eukaryotes. Reason: both bacteria and yeast multiply very fast to form huge population to express the desired gene. That is also true. So our answer will be both the assertion and reason are true, and reason is the correct explanation of assertion. Okay. Now let's jump to the next topic, that is the tools of recombinant DNA technology. So basically, what are the subtopics we're going to study? We'll talk about restriction enzymes, ligase enzymes. So when I'm talking about restriction enzyme, they come under the group of nuclease enzyme. So if we talk about nuclease, if you want to try to link with the biomolecule chapter, the enzyme section, there are 6 classes of enzymes are there: OTH-LIL, oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. This is the first class, second class, third class, fourth class, fifth class, sixth class. So question can be asked, restriction enzymes belong to which class of enzyme? So they belong to the hydrolase or the third class of enzyme. They perform hydrolysis of sugar and phosphate backbone, right? So they are hydrolase. If someone asks you what about ligase, they belong to the sixth class of enzymes. They're responsible for pasting to different strand of DNA or pasting up complementary DNA strands. Okay, now we need help of tools like gel electrophoresis. Gel electrophoresis is responsible for separation of DNA fragments based upon their size. We need the help of cloning vector. Cloning vector, they act as gene taxi or a vehicle to transfer gene from one organism to another organism. And we need to learn how to make the hosts competent enough so that its insertion of the DNA for the transformation will be successful, right? So let's start with restriction enzymes. Okay, so first we'll talk about the discovery of restriction enzymes. So when I'm talking about discovery of restriction enzyme, it was the year of 1963, and the scientists, we must talk about them, Smith, Arbor and Nathan. They got Nobel Prize as well for this revolutionary discovery. Boyer was expert of restriction enzyme but discovered by Smith, Arbor and Nathan, right? Okay, so what they found, so they were checking in detail about this bacteria called E. coli, Escherichia coli. So they found uh, to counterattack the viral infection, this E. coli bacteria, they have two very special enzyme produce them. So what they found, to counter or to inhibit that, to counter the viral attack, E. coli have two enzymes. E. coli have two enzymes. Okay. Now, what are the utility of these two enzymes? One enzyme that is responsible for the methylation of bacterial DNA to prevent the viral integration. So, one enzyme responsible for methylation of own DNA, means bacterial DNA, and the name of the enzyme was methyltransferase. Basically, they transfer methyl group, they transfer methyl group within the nitrogenous base cysteine. And if the nitrogenous base is methylated on DNA, so the virus or the viral DNA will be unable to integrate it within the cellular genome. So that is the strategy taken care of by methyl transferase. Okay. Now, what is the second enzyme? So basically that cleave or cut viral DNA into pieces, right? One is if this is the bacteria, and if this is the bacterial genome, suppose this is the bacterial genome, the methylation is taking place so that the virus which is attacking the bacteria, basically the bacteriophage virus, they insert their genome into bacteria. So this methylation is inhibiting the integration of viral DNA, vDNA, with hDNA, means host DNA in shortcut. vDNA, viral DNA, hDNA means host DNA for our example. Okay. So that is the function of methyl transferase. What is the function of the second enzyme which can cleave or cut viral DNA? We call it restriction endonuclease. Restriction endonuclease that cut the viral DNA, by recognizing their specific site into different pieces. So when the viral DNA is cut into different species, they can't integrate themselves with host DNA. And if there is no integration, there will be no transcription and there is no translation. If there is no translation, there will be no formation of the parts of virus. So this is how the immunity of prokaryote works, right? Okay. Now, so this was all about the discovery of restriction enzymes. Okay. Now we'll talk about the type. No, first we'll talk the restriction enzymes and their classification. Yes. So basically, there is a special class of enzyme called hydrolase. They are responsible for hydrolysis, and one subclass of hydrolase we call it nuclease. Nuclease means that has the cleaving property to RNA or DNA. So nuclease basically it can produce cut either to nucleic acids, RNA or DNA. So if it's cutting RNA, we will call it ribonuclease. If it's cutting DNA, we call it deoxyribonuclease. Okay. Now again, the nuclease, that is of two types. One is exonuclease and another is endonuclease. Exonuclease and endonuclease. Now, what is the difference between exonuclease and endonuclease? Again, this is the molecular scissor, suppose, and this is the piece of DNA. If the molecular scissor is cutting DNA from the edge, if cutting DNA from the edge of a DNA molecule, DNA strand, that is called exonucleus, and if the molecular scissor is cutting DNA from inside, and that is called endonucleus. I think it's clear now. So exonucleus, they cut DNA from the edge but endonucleus cut DNA from inside. So that is the basic difference between exo and endonucleus. Okay. Now while I'm talking about this endonuclease enzyme, when I'm talking about the endonuclease enzyme, they randomly inspect the length of DNA. So suppose this is the one of the endonuclease enzyme. Randomly they will inspect, they will read the alphabet, genetic alphabet of DNA. They will read the sequence of DNA and they will search for special recognition sites. So how this endonuclease work? They inspect the length of DNA. First they inspect the length of DNA. After that they recognize special sequence we call this special sequence as recognition sequence. And majorly this recognition sequence, they are palindrome. They are palindromic sequence. So what do you mean by palindromic sequence? Some of the example of palindrome I am writing here. Suppose if I write the word Malayalam. So if you study from here, if you read it from here, M-A-L-A-Y-A-L-A-M, and from back side to front side, M-A-L-A-Y-A-L-A-M. So from this direction and this direction, in both the direction, they have similar letters type. Another word, madam. From this side, madam, from this side also madam. Another word, dad. From this side, dad, from this side also dad. So these are some common examples of palindromic sequence, right? But in case of restriction enzymes, REN, full form of REN is restriction enzymes or restriction endonuclease. They will search for a special palindromic sequence. So here you can check it for if the restriction enzyme is EcoR1, it will search for the palindromic sequence called GAATTC. If the restriction enzyme is Hind2, it will search for the restriction sequence GTTAAC. So from 5 dash to towards the 3 dash, if you read it like that, GAATTC, and from again to the, as because the strands are anti-parallel, so you have to read from the 5 dash to 3 dash. One is 5 dash to 3 dash, one is 3 dash to 5 dash. So if you go from 5 dash to 3 dash, you will get GAATTC. If you read from the end for the anti-parallel strand from 5 dash, again it will be GAATTC. So this is the importance of palindromic sequence. Now if restriction endonuclease is EcoR1, it will search for GAATTC. If the restriction endonuclease is Hind2, it will search for the palindromic sequence of GTTAAC. I think this is clear now. Okay. Now further information. Now when you are talking about restriction endonuclease, if we talk about the types of restriction endonuclease, there are two types. One is type 1 restriction endonuclease, another is type 2 restriction endonuclease. Now what is difference between type 1 and type 2? Type 1 restriction endonuclease, they have dual function. They can methylate, they act as methylation, they also act as cutting. They cut and methylate. So they can perform like methyl transferase as well as restriction endonuclease. That's why I am telling they have dual function. But type 2 restriction enzyme, they can only produce cut by recognizing palindromic sequence. Okay. And also in higher class you will learn that there will be difference between the cut produced by type 1 restriction endonuclease and the cut produced by type 2 restriction endonuclease. Okay. Basically type 1 restriction endonuclease, they will cut far from the recognition sequence, but type 2 restriction endonuclease, they will cut either within the restriction site or near to the restriction site. Okay. So our restriction endonuclease, the type we are talking about, that is type 2. Type 2, this is used in genetic engineering. Type 2 restriction enzymes, they are used in genetic engineering, right? Okay. Now again, this type 2, they can produce sticky end cut, they can produce blunt end cut. So both the case is possible for type 2 restriction endonuclease. Now what is the difference between sticky end and blunt end cut? If this is the DNA sequence, so one restriction enzyme will cut directly from the center, at the center of recognition sequence. So that will produce blunt end cut. So this is how the DNA strands will look like upon blunt end cut. So this cut is blunt end, but another way is there to form sticky end cut. Already we have discussed, it will produce the overhanging stretch. So the overhanging stretch when it's produced, we call it sticky end cut. So which is easier for our purpose? Sticky end cut is more easier. It will give more advantage while you want to construct a recombinant DNA, right? And you need to remember that while you are producing sticky end cut, they don't produce cut just exactly at the center of the palindrome. Suppose if this is the palindrome GAATTC, GAATTC, they will not produce the cut exactly at the center of palindrome. They will produce cut near to the center, like they will produce cut in between G and A. If I am talking about eco R1, that will produce a sticky end cut, but we are talking about another restriction endonuclease, that is hind 2, that produces blunt end cut. Okay, few more thing about restriction endonuclease. First discovered restriction endonuclease is hind 2. This is the first discovered restriction endonuclease. Next, in restriction endonuclease used by Boyer and Cohn, that is eco R1. You need to remember this. Hind 2, first discovered restriction endonuclease. Now you can ask me, sir, why hind 2? Why not hind 1? Okay, for that you need to know the process of nomenclature of restriction endonuclease. So now let's talk about nomenclature. Nomenclature of restriction endonuclease. Okay, so let's take any name, example of eco R1 and hind 2. Let's nomenclate them. So the first letter is about the genus of the bacteria. First letter is the genus of bacteria. So you need to know that more than 900 RANs, restriction endonuclease, are isolated from 230 strains of bacteria. Very, very, very important information. How many? More than 900 restriction endonuclease isolated from 230 strains of bacteria. Statement based questions can be directly asked. So we are talking about the first letter. Now let's talk about the second and third letter. Okay, so for E. coli, Escherichia coli, the E letter we have taken from Escherichia. Is the generic name. If we talk about H, it is for Haemophilus. Okay. Now, second and third letter is species name. Taken from species name. For example, co from the word coli, in for influenzae. So the first two letter of species name is taken for the second and third letter. Okay. Now the fourth letter. You need to remember this fourth letter is optional. In the process of nomenclature, it's not mandatory to keep the fourth letter. But as we can see here, R and D as fourth letter of echo R1 and hind 2. So fourth letter means the strain of bacteria, right? For example, RY strain of E. coli bacteria is responsible for the letter R. RD strain of Haemophilus influenzae is responsible for the letter D. Okay. Now fourth, a Roman number is given here. What is the importance of this Roman number? The importance of this Roman number echo R1 and hind 2 is the order of isolation. Order of isolation. It's not the order of discovery. Remember this. Like after how many trials or from which lab it was extracted. So here echo R1 is the order of isolation, hind 2 again the second order of isolation. So that's why hind 1, why not hind 1 is the first discovered restriction endonuclease. Before hind 2, hind 1 was isolated. Before hind 2, hind 1 was isolated. But the function of proper restriction endonuclease was identified inside hind 2 enzyme. So that's why we say that this is not the first isolated restriction enzyme, but first discovered enzyme as which has the function of restriction endonuclease. Remember it. Okay. So we are completed with this topic. Okay. So the third point where I said that though the restriction enzyme, if we talk about echo R1, if we elaborate the nomenclature method, we'll get Escherichia, then coli, then the strain is RY13 strain, and the one is the order of isolation. Okay, but here I said that this fourth letter is not mandatory. There are certain examples like Sal 1, PST1, PVU1. So these are some examples of restriction endonuclease where you won't be finding the fourth letter. So that's why I was telling that it's not mandatory to have the fourth letter in the name of restriction endonuclease. And one more thing you need to remember, if you want to construct rDNA, so there is, if this is one of the DNA piece and this is another DNA piece. So here you have got the gene of interest. So you want to cut it and transfer the gene of interest inside this particular DNA. There are two DNA molecules, DNA1 and DNA2, and you want to construct rDNA, right? So you have to use for the cut of these two different DNA, you have to use same restriction endonuclease. So if you are cutting this DNA with ECO R1, you have to cut this DNA as well with the same ECO R1. If you are cutting this DNA with BAM-H1 restriction enzyme, you have to cut this DNA with the same BAM-H1 restriction enzyme. If the sticky end is not produced proper manner, then how you just try to think that how you can produce a proper recombinant DNA, right? So that is the reason you need to cut two different pieces of DNA, and if you want to recombine them in future, the cut should be in a proper manner with the help of same restriction enzyme. Do not forget it. Okay. Now, what after restriction in the enzyme? We'll talk about the enzyme ligase, responsible for ligation or pasting different pieces of DNA together. So basically the pasting enzyme is called ligase enzyme. Pasting enzyme is called ligase enzyme. So you have taken one piece of DNA, you have another piece of DNA as well. Now within this DNA you have your gene of interest. This is the span of your gene of interest. So what you have to do, you have to produce cut here as well as you have to produce cut here. So here a sticky end cut will be produced. Suppose this is the gene of interest. So in the similar way, here also sticky end cuts will be produced. Now what you have to do, you have to take out the gene of interest. You have to take out this gene of interest from here. and you have to put this inside the vehicle DNA. So that's how it works. Okay, so that is the importance of sticky and cut, and for pasting purpose the enzyme that is used. So this is a cut piece of the gene of interest, and this is the cutting end of the piece of DNA where you want to insert this. And how to insert this? You need a paste, biological paste, and for the paste your enzyme ligase will work here. Now after the action of ligase, you can say successfully you have converted a simple DNA into recombinant DNA, right? Okay. Now we learn the process of gel electrophoresis. The process of separation of DNA according to their size. Now before pasting this space of DNA, once you are done with the function of restriction enzyme, so in that medium you have random pieces of DNA. Now what you have to do, you have to separate out the gene of interest from this random piece of DNA. So how to separate the gene of interest from other large pieces of DNA? By the process of special process of separation, and we call it gel electrophoresis. By the process of gel electrophoresis. So this is the process of separation of DNA strands, DNA molecules according to their molecular weight, according to their size, right? How to determine the size of DNA molecules? It can be 10 base pair, 100 base pair, 1000 base pairs, right? So these are different size of DNA. So which is smallest one? 10 base pair DNA. Which is the largest one here? 1000 base pair DNA. So you can separate out DNA of different size. Okay. So to perform this gel electrophoresis, what you need? You need a gel plate. You need to make a gel plate. From where this gel we are getting, the gel, the source of this gel is basically, we call it agarose gel, which is obtained from seaweeds, specifically red algae, Gracilaria, Gelidium, right? So from red algae we need to extract agarose gel. So the agarose gel is used to form the gel plate. Okay. Now what you have to do? Suppose this is the gel plate. You need to connect a machine. One is positive electrode and another is negative electrode. One is cathode and one is anode. The positive electrode, that is anode, the negative electrode, that is cathode. Now here what you have to do, you have to keep some wells where with the help of micropipette you will put this DNA molecules. Right? Now switch on the circuit. Now as because DNA molecules, when you talk about DNA, that is negatively charged, DNA molecules are negatively charged, right? So the positive electrode will attract DNA molecules. DNA molecules have enough anions or negatively charged ions. So anions will be going towards anode or positive electrode. Remember this. So how they are moving from negative electrode to anode? When you are talking about this agarose gel, there will be tunnels, different tunnels of different size present inside the agarose gel. So they will act as sieve. So this sieving property, of agarose gel will promote movement of smaller DNA molecules faster and will inhibit movement of larger DNA molecules. So large DNA molecules will move slowly, small DNA molecules will move fastly. Suppose in a running competition, there are few like thin members are there and few fat members are there. So who will run fast? Obviously thin members. So similarly, the smaller DNA molecule, they will move really fast. And the larger DNA molecules, they will move slowly. So this is how the different DNA molecules are separated based upon their size. Now it's time to identify and take them out or isolate them out. So basically what we have learned now, which property of agarose gel helped us to separate out DNA based upon their size? Sieving property of agarose gel. Inside the agarose gel plate, there will be microscopic tunnels through which DNA molecules will move forward towards the anode. Remember, DNA negatively charged, DNA molecules are negatively charged, they are acting as anions. Anions means negatively charged ions. They will move towards positive electrode or anode. Okay. Now, with the help of our naked eye, we cannot observe these bands of DNA. So for that, what you have to do? You have to apply a stain. Which stain is applied here now? You have to apply the stain called ethidium bromide, ETBR. Ethidium bromide is applied on this gel plate. You have to make a solution of ethidium bromide with the help of brush. You just have to paint the gel plate. And under ultraviolet rays, under UV rays, under ultraviolet rays, the DNA fragments will give fluorescent color, will illuminate as orange color. So orange illumination can be observed. So wherever in this gel plate you can observe this orange luminescence, that means that signifying the presence of DNA molecules. Okay. So if you can see it, this is the gel electrophoresis, the machine where we perform gel electrophoresis, right? This is the site where you will find a well. You have to load the DNA molecules. This is the side of cathode, and this is the side of anode. Remember it. And this is the gel plate. And here you can observe this luminescence on gel plate. They're denoting the presence of DNA strands, right? Okay. So we are done with this process. So here also you can observe how the whole gel electrophoresis process is performed. So after we have identified, suppose we have to isolate, only this pieces of DNA. So after that, what you will do, the process will be scooping and elution. So after that, the process will be scooping and elution. Now what is that? What is scooping and what is elution? With the help of spatula, we'll cut out, like you cut your birthday cake, we'll cut these pieces, right? So that process is called scooping. Like with spoon you are taking a scoop. And now the next process is the process of elution. So here we need a buffer medium. Now what this elution is doing, elution is the process by utilizing a elution buffer, we will purify, or we will take out the DNA strand, we will separate them out from the agarose gel. Separation and collection of DNA strands. So that is the process of elution. Okay. Now a question can be asked that, sir, for the purpose of staining, why we use ethidium bromide? Why not other stain? Because under ultraviolet rays, basically the ethidium bromide molecules, they will get trapped inside the DNA molecule and they will absorb the wavelengths of light and radiate the orange color. So that can be easily visible. So for the visibility, of DNA, we use ethidium bromide because DNA strand cannot be visible with the help of our naked eyes. Remember, with your naked eye, you cannot observe the small DNA strands inside the agarose gel plate. Okay, now we are done with restriction endonuclease, we are done with ligase, we are done with gel electrophoresis. Now we'll jump into one of the most important topic of this chapter, that is cloning vectors. So let's talk about cloning vector. So let's talk about cloning vectors. So cloning vectors, they act as taxi or vehicle to deliver the gene of interest inside the host organism. That is called cloning vectors. Yes. Now, we'll talk about different types of cloning vectors. There will be cloning vectors for bacteria. If you want to transform a bacteria, then what kind of cloning vectors you should use? You want to transfer a plant cells, what kind of cloning vectors you should use? And there are certain other methods where we will not be utilizing vectors to transform a recombinant DNA inside our host organism or a host cell. Okay. So, for bacteria, the cloning vectors are used. They are mainly plasmids, or bacteriophage. They are majorly used cloning vectors to transform a bacteria. Apart from them, you can use cosmids, you can use plasmids, you can use bacterial artificial chromosomes. Okay. If you want to transform a plant cell, what kind of vector you should use? For transforming a plant cell, specifically to transform a dicot plants, the vector that is used, that is called agrobacterium or natural genetic engineer. Agrobacterium tumefaciens. A bacteria is used, and from this agrobacteria will that infect the plant, and that is responsible for abnormal growth or cancer. That has a special plasmid called TI plasmid, right? This TI for tumor inducing plasmid, so that causes cancer of plants. So that's why agrobacterium is also called as natural genetic engineer, and they can deliver the this special DNA present in TI plasmid. TI plasmid has a special DNA called tDNA inside the plant cell, right? So that's why we call it natural genetic engineer. Now you can ask me, sir, so we can transform a dicot plant cells with the help of agrobacterium. What about monocot plants? So in laboratory, few strains of agrobacterium nowadays we can transform even monocot plants. And for normalized generalized technique, there will be other techniques, non-vector mediated techniques are there to transform plant cell or to deliver gene of interest. We can use biolistics or gene guns. Okay. So let's talk about plasmid. We'll try to means exaggerate, or our will be describes our description will be mainly upon plasmid. Sorry for the fumbling. Okay. Let's have some water, then I will continue. Okay, so let's elaborate about plasmids. So if we check a bacterial cell, so this is a bacteria and this is the chromosome. Means bacteria, they have one single chromosome, circular DNA, that is also can be called as nucleoid, right? Now apart from this chromosome or nucleoid, if you find ringlets inside the bacteria, and if these ringlets are self-replicating, so what is plasmids? Plasmids are self-replicating extra chromosomal, they are not part of the main chromosome, circular double stranded DNA ringlets. So they can replicate, produce their own copy by their own. Yes. So you got the definition of plasmid, right? Now the bacteria during the time of conjugation, they can pass these plasmids to another bacteria. Okay, what we call the sexual reproduction in bacteria. Actually, the process is called conjugation. But here we will be utilizing this plasmid as our gene taxi. Okay. So there are different types of plasmids are there. High copy number plasmid and low copy number plasmids. Like if you find 1, 2, 3 copies of plasmid, sorry, if you are finding 10 to 100 copies of plasmid, that is called high copy number plasmid. If you are finding 1 to 3 copies of plasmid, then we call them low copy number plasmid, right? Now what are the factors behind this high copy number? Why few plasmid can produce multiple copies? Why few plasmids can produce only few copies? We'll discuss about that. Okay. Now why we are choosing, or which kind of plasmids or vectors can be chosen in biotechnology or genetic engineering? So three criteria are there. So the criteria you need to get inside a vector or inside a plasmid. Okay. Three features. Three features which qualifies them for a fantastic gene taxi. For the qualification of to work as a vector or gene taxi, what are the three features you need to get inside a vector or plasmid? Okay, the first criteria is presence of origin of replication. Second criteria, presence of selectable markers. Third criteria, cloning site. Cloning or recognition sites. origin, presence of selectable marker, presence of cloning site or recognition site. Now, what are the importance of origins of replication, also called as ori site? So, these are special sites of plasmid. These are special sites of plasmid which is rich in adenine and thymine. So there are, if inside the DNA, if you peep inside the DNA, then what are the base pairing sequence you are gonna get? A will bind with T, adenine to thymine, and G will bind with C, guanine to cytosine. But A binds with thymine with double bond or two hydrogen bonds, and G binds with cytosine with triple hydrogen bond. So while the replication fork will be produced, you need to you need to separate two chains of DNA. Now, the more stronger bond will be there in between them. It will be harder for you to separate them. But if the bond is quite lighter, you can easily break the bonds. So it will be an easier task for you, so that you can separate out two different DNA strands. So origin of replication is the site where replication bubble or replication fork is produced by separating the two antiparallel DNA strand. So that's why it's rich in A and T. So more double bonds are there, so it will be easier for you to separate them out. If there is more triple bonds, it will be harder for you to separate them. Okay, so that is the thing that is rich in AT. This is the site of initiation of DNA replication. So ORI site is the site of initiation of DNA replication, and also you need to remember the ORI site that regulate the copy number of a plasmid. ORI site that regulates the copy number of a plasmid. So if during the time of initiation of replication, if it's easier to open the chains, so gradually there will be tendency to produce high copy number. So if it's harder to open the chains, so there will be tendency to get the low copy numbers for your plasmid. Okay. Now selectable markers, these are also called as reporter genes sometimes. So basically, the presence of these selectable markers, they will report us, they will give us signal, they will help us to identify or confirm either the cell is transformed or not. Okay, so selectable markers helps in identification of transformants and non-transformants. We can differentiate by observing the presence of these reporter genes or marker genes, either the colony of bacteria are transformed or not. Okay, so as selectable marker, what kind of genes are preferred? As selectable marker, you can prefer the antibiotic resistant genes like ampicillin resistant gene as AMPAR, tetracycline resistant genes as TETR. Okay, but sometime it will create problem for you to make the job more precise, sometime apart from this antibiotic resistance gene, we'll be utilizing other genes, that is lag Z. Okay, we will explain in detail when we'll use these antibiotic resistant genes and when we use lag Z gene. Now, what is the importance of cloning sites or recognition sites? Now, in order to construct a stitch or paste to form a new DNA, recombinant DNA, you need to cut and stitch, right? So, where to cut, where to stitch, where to paste? So, you need that particular site where restriction endonuclease will cut the DNA sequence and ligase will paste the DNA sequence, DNA strands. So, that is the site of cloning sites or recognition sites. So, basically they are nothing but sites, they are restriction sites, the restriction sites for RAIN, restriction endonucleases. And one more thing you need to remember this, for a single plasmid, if you talk about one plasmid, it's preferable that only one restriction site is present for one restriction enzyme. Remember this. One restriction, if you are talking about the restriction enzyme BAM-H1, so there should be only presence of one BAM-H1 recognition site. If there is two BAM-H1 recognition site, so the same enzyme BAM-H1 will cut the plasmid into two different pieces. So that will be a more complex or hideous job to perform while you are pasting them with the help of ligase. Okay, so let's talk about the first artificial plasmid which is used in genetic engineering. So name of that plasmid is PBR 322. Okay, so what about this particular plasmid PBR 322? There is plasmid P for plasmid, B for Bolivar, R for Rodriguez, and 322 is the lab number. Okay, so let's construct this plasmid PBR 322. So this plasmid has two selectable marker site, that is two antibiotic resistant gene is present into this. One is ampicillin resistant, another is tetracycline resistant. And apart from these two sites, they have two different sites. One is ori site or origin of replication site, and another is ROP site or repressor of polymer site. Yes, repressor of polymerase, ROP site. Yes. So this is our PBr322 plasmid. This is the ampicillin resistant gene. And this is the tetracycline resistant gene. Okay, now I'll talk about the presence of marker. Okay, now one more thing, this is the Ori site and this is the repressor of polymer site. On ampicillin resistant, there will be recognition site for PVU1, PST1. On rope site, there will be recognition site for PVU2. On tetracycline resistant gene, there will be two recognition site for two different restriction endonuclease, SAL1 and BAM-H1, and in between this stator and amper, there will be restriction site for HIND3, CLA1 and EcoR1. These are different restriction sites. So let's sum up that the structure of PBr322. It has a Ori site or origin of replication. It has one rope gene that is called repressor of polymerase. So basically both these genes, they regulate the copy number. Ori site is responsible for producing more copy number, and a rope gene that is responsible for repressing, inhibiting the copy number. So these two genes are responsible with the copy number of this particular plasmid. Okay. Now when you are talking about the selectable marker, so it has two selectable markers. One is ampicillin resistant gene and another is tetracycline resistant gene. If I talk about like how they work, they stop the attack of antibiotic if this plasmid is present inside a bacteria. So we use antibiotic to kill the bacteria. Now suppose you have administered ampicillin. Now as because it has the AMPAR gene, so what will be the product of the AMPAR? It will be beta lactamase. Beta lactamase. So this beta lactamase that will nullify or neutralize the effect of ampicillin antibiotic. So this is how they stop the attack of antibiotics. So they are boon for bacteria which has this particular plasmid. Okay. Now if we talk about the cloning sites or recognition sites to produce cuts. So from ampicillin resistant, you will get PVU1, PST1. Then on rope you got PVU2. On TET-R you got SAL1, BAMH1, and after that you will get Hind2, Hind3, CLAV1 and ECO R1. So these are different sites for different restriction endonuclease. PVU2, this is the name of restriction endonuclease. It can produce cut on rope gene. If you are using BAMH1, that will produce cut in tetracycline resistant gene. If you are using CLAV1, that will produce cut in between tetracycline and ampicillin resistant gene. Right? If you are producing PST1, that will produce cut on ampicillin resistant gene. Okay. So I remember in this way, pav bhaji, PVU1, you are eating something, PVU, pav bhaji, PST, pasta. So pav bhaji, pasta, pav bhaji. So you have eaten two pav bhaji. PVU1, first pav bhaji, PST1, pasta, PVU2, second pav bhaji. सलाद BAMH1 बना के, पाव भाजी पास्ता पाव भाजी सलाद बना के हिंदी क्लास में इकोनॉमिक्स, हिंद 3, CLAV1, ECO R1. So it's like that. Again, this is a mnemonic for to memorize the cloning sites on PBR 322. पाव भाजी पास्ता पाव भाजी सलाद बना के हिंदी क्लास में economics. Is it okay? Great. So now how to utilize these plasmid of Bolivar and Rodriguez to transform E. coli bacteria? Let's study that. Okay. So suppose you have inserted a gene of your gene of interest within the BAMH1 site. So what will happen? That will cut the BAMH1 site, the BAMH1 enzyme. First you have to cut the BAMH1 using restriction endonuclease BAMH1. You have to produce a cut here. Now once you are inserting the gene of interest into this BAMH1 site, the functionality of this TET-R gene has gone because you have disturbed already. So it cannot produce the mRNA and protein. So it cannot stop the attack of tetracycline antibiotics. So it is no more tetracycline resistant. Now if you are forming a recombinant DNA by putting a cut here into the BAMH1 site, right? Now if you are forming a recombinant DNA by putting a cut on PST1 site on AMPR, then again what will happen? It has the tetracycline resistant gene, but the AMPR has gone. You already cut the AMPR, the functionality will not be exhibited. It is no more resistance against the ampicillin enzyme, right? So let's perform one experiment that we call insertional inactivation. Insertional inactivation. So what do you mean by this? Upon inserting the gene of interest, upon inserting the gene of interest in a cloning site, means recognition site, within the selectable marker, within the antibiotic resistant gene, Upon inserting the gene of interest in a cloning site within the antibiotic resistant gene, the resistance against the particular antibiotic is gone. So that is called insertional inactivation. For example, we were talking about this PBR. So suppose it has two site, one ampere and another is tet R. Now what you have done, you want to insert your gene of interest into this BAM-H1 site by utilizing this BAM-H1 enzyme. So this is your gene of interest. So this is your gene of interest. You have inserted into this, into PBR. Now though it has intact ampicillin resistant gene, but the tetracycline resistant already got splitted, so it will not show effect of tetar because you have your GOI, gene of interest, here. So the tetracycline resistance is gone. So we will be saying that this plasmid is inactivated for tetracycline resistance, or the selectable marker will not work here, right? Okay, so let's focus on bacterial colony. How to, now you are culturing a bacteria into your lab, so how to identify which colony is transformed, which colony is not transformed, right? So let's study that. Suppose after performing everything, this plasmid, you are gonna insert into E. coli bacteria, right? So for that, all the processes you will perform, you will be making E. coli competent. Now after that, the E. coli are formed. These are different E. coli bacteria, the E. coli colony. Now you wanna insert this into each and every bacteria, but it's not always it will occur that the bacteria will happily intake this plasmid. Okay, at some time that is also one of the possibilities there. After performing this rDNA technology, maybe your plasmid is reluctant to intake the gene of interest, so it will remain as it is. So it will remain non-recombinant, right? So there are two types of plasmid. One plasmid which has taken the gene of interest, we call that recombinant, and the plasmid which has not taken the gene of interest, we will find the intact AMP R and tetar gene because the gene is not inserted, that is called non-recombinant plasmid. So we are trying with thousands of plasmids into your lab, but not all the plasmids will take the gene of interest, and after that, while you are trying to transform the E. coli bacteria, not all the bacteria will take the plasmids. So there are three possibilities. One possibility is non-transformant. Now what do you mean by non-transformant? The E. coli which has not taken any of the plasmid, either recombinant or non-recombinant. Okay. Now what are the next possibilities are there? Next possibility is there that few E. coli bacteria, they have taken the plasmid, but they have taken the non-recombinant plasmid. So what we will call them? Non-recombinant transformants. And there are few E. coli, they are really good for us. They have taken the rDNA or recombinant plasmid. So we will call them recombinant transformants. Right? So upon the experiment of transformation, you want to transform all the bacteria, but the chance is sometimes the chance is not 100% the bacteria will intake the plasmid. So if there is no plasmid, it has failed to intake the plasmid, then we will call this bacteria as non-transformant. It has taken the plasmid, but not the plasmid with gene of interest, the plasmid without the gene of interest, the non-recombinant plasmid, then we will call this bacteria as non-recombinant transformants. And the bacteria, our own bacteria, we will like this bacteria, this is our friend, they have taken the recombinant plasmids, so they are called recombinant transformants. So there are two types of transformed bacteria are produced, non-recombinant transformed and recombinant transformed. Now we will try to learn how to separate out the non-transformants from transformants. It's very easy. So inside the petri dish, so this is the petri dish, you are culturing them. This is the petri dish, you are culturing them. This is the culture media. And here you will find the colony of bacteria. So there are three colonies, right? You can observe three colonies. Colony A, colony B, colony C. Now, from watching outside, by observing outside, you cannot identify which one is transformed and which one is not transformed, right? So, what you can do now, you can apply, suppose you have applied ampicillin, antibiotic on the petri dish. Now, what will occur after application of ampicillin? You will observe that colony of B and C, colony B and C, they are alive, but colony A has eliminated. So the death of colony A means they are the colony of non-transformed bacteria. So here you can infer that A, they are non-transformed. That's why they have died while you have applied ampicillin. Upon administration of ampicillin, they will die because B and C, suppose they are non-recombinant transformant and recombinant transformant. They both have the plasmid, and both the plasmid, they have ampicillin resistant gene. So upon application of ampicillin, no one, either it's non-recombinant or recombinant, they will die. Okay. Now you have to separate out the recombinants from the non-recombinants. So B and C, they both are transformed. They have the PBr322 plasmid, but you are not sure which one has the recombinant PBr322 and which one has the non-recombinant PBr322. So what you will do now? The next thing you have to do, you have to administrate tetracycline. You have to administrate tetracycline antibiotic. Now, after that, what you will observing inside the petri dish, suppose only colony B has survived, C has also gone. Tetracycline has killed colony C. So, what will be your concluding statement? So, colony B, these are the recombinant, these are the non-recombinant transformants. As because this non-recombinant transformant, they got the intact PBr or non-recombinant PBr322 or non-recombinant plasmid. But colony C, they are of recombinant transformant because due to the insertional inactivation, the tetracycline gene has vanished. So colony C, they are recombinant transformants. But again, if you think you need to isolate the recombinant transformants, right? You need to work with recombinant transformants. But if you are doing so, whatever we have done here, we have lost the colony C that are the recombinant ones. So again, it will become a hideous job or complex job for us. So what you can do, before performing the application of tetracycline on the petri dish, we have to perform another task that is called replica plate method. We have to perform a replica plate method. With the help of another petri dish, we have to separate out the impression of colony B and colony C. Like impression of colony B and colony C. So after administration of tetracycline, when only colony B is survived but colony C has gone, still we have this replica plate with us. Into this particular zone of this plate, we have our recombinants. So it's a quite complex task. So that's why scientists, they started thinking, we want a precise method. Instead of utilizing these antibiotic resistant genes as selectable marker, let's find some other gene which will help us to identify the recombinant transformants. So for that, they used another plasmid called PUC19. So in this plasmid, they got another gene that is called LAC-Z gene. Okay. Now LAC-Z gene, this is not a ampicillin tetracycline antibiotic resistant one. So how it works? The LAC-Z gene, the product is beta galactosidase. Now, beta galactosidase, what is the utility of this beta galactosidase? How we can identify that if one bacteria has this LAC-Z gene or not inside it? So, beta galactoside, when artificially you will supply into the culture media, you will supply the substrate called X-gal. We call it chromogenic substrate. So beta galactosidase, that is an enzyme, so that needs a substrate, and if the substrate is X-gal, which is a chromogenic substrate, when the enzyme will bind with X-gal and it will produce the product, it will form a blue color, a blue colored colony. So if you are culturing some E. coli bacteria with a plasmid which has the LAC-Z gene, and if you are applying into the culture media, if you are applying the chromogenic substrate X-gal, so they will produce blue colored colony. Okay. Now, how it can be one of the replacement method of antibiotic resistant gene? So, suppose this is the plasmid. Suppose this is the plasmid. Here we have one antibiotic resistant gene associated with it. Suppose this is AMP-R gene, and suppose here we have this LAC-Z gene. So this is not present, the site of cloning is also present inside the LAC gene, LAC-Z. Now you don't have to cleave the sites present inside the ampicillin resistant gene. Now if you are cleaving or if you are inserting the gene of interest into the LAC-Z, then what will happen? The recombinant plasmid that already have these antibiotic resistant gene, ampicillin resistant gene or AMPR, and now you have what you have done, you have inserted gene of interest in between the LAC-Z. So when the LAC-Z is splitted, so what you will do, it cannot produce beta galactoside and the colony will not be producing color. So now this become the recombinant plasmid. Now cannot produce, when you are inserting gene of interest inside the LAC-Z, the bacteria cannot produce blue color. So the colony on petri dish, they will not resemble blue. So if you have inside your culture, bacteria culture, you have one blue colony or two blue colonies there and one white colony or colorless colony, that means these blue colonies are non-transformant or non-recombinant colony, and the colorless colony, that is recombinant colony. Colorless colony is the recombinant colony with the recombinant DNA or rDNA. So that is the alternative use of selectable marker other than utilizing antibiotic resistant gene. So it is clear, is it clear? So recombinant here, non-recombinant will be blue in color, but the recombinant will be colorless. Remember this. Generally student will do mistake. They thought that blue color colony means recombinant colony and colorless colony means non-recombinant colony, but it's opposite. Recombinant colony means laxate is splitted by the process of insertional inactivation. Okay, so we are done with vector. Okay, now let's talk about some other vectors. We were talking about the Agrobacterium tumefaciens. Already I think we have spoken. With the help of that plasmid called TI plasmid, we can insert gene of interest into dicotyledonous plants, right? You just have to produce a genetically engineered TI plasmid. Within the tDNA, there was a DNA piece inside that TI plasmid called tDNA. So what you have to do here, this tumor causing DNA, you have to insertionally inactivate it. You have to put the gene of interest within the tDNA so that the TI plasmid now will become harmless or disarmed. Now this harmless TI plasmid can be inserted into any dicot plant cell. Okay, now one thing you need to remember this TI plasmid that act as episome. Now what do you mean by episome? That can directly transfer. We are not talking about prokaryote here. This triplasmid are vehicle for eukaryote cell, plant cell. So what they will do, they will directly transfer the gene of interest into the plant chromosome. That is called episome. Transfer the GOI into the plant chromosome. So this special property of special plasmid. Okay, now how to transform an animal cell? Or vector for animal cell? So we know that generally retrovirus means they're basically RNA virus. They're very much efficient to attack animal cell and transfer the DNA, the viral DNA inside the animal chromosome. One of the example is human papilloma virus, HIV, you know very well. So there are certain other retrovirus as well. So what you have to select, you have to select a less virulent retrovirus. You have to genetically modify it. So that genetically modified retrovirus, will be calling them disarmed pathogen or disarmed retrovirus. Now with the help of this disarmed retrovirus, if we infect animal cell, this is the retrovirus. So what they do, they will inject their RNA, and this RNA will be converted by the process of reverse transcription into viral DNA. That's why we call them retrovirus, from RNA to DNA conversion. And now this DNA is directly incorporated with host chromosome, right? So we'll be utilizing this strategy. So upon infection of animal cell, the gene of interest is transferred into animal cells to make them transformed. So remember, for bacteria, the vectors are used commonly. They are either bacteriophage or plasmid. For dicot plant, Agrobacterium tumefaciens, we got the plasmid from Agrobacterium tumefaciens. The name of the plasmid is TI plasmid. And to transform animal cell, generally we can use disarmed retrovirus, genetically engineered retrovirus. So they will infect, but they will not cause disease. Rather, they will transfer the gene of interest into our genome. Okay. Now, we'll talk about some other methods. So now we'll talk about some other method to transform the host cells. Okay. Transforming host cells. So we already know about the method that is majorly used, that is vector mediated transformation, vector mediated, and another method where there is no use of vector. Okay, non-vector mediated. Already we are done with vector mediated method. Now let's talk about non-vector mediated methods of transforming cells. Yes. So for bacteria, the methods are used, that is use of cold calcium chloride with calcium ion along with heat shock. Okay, so that we call the process of making bacterial cells competent. So how to make bacterial cells competent and why it's required? So it's required because if you simply offer a bacteria, suppose this is the bacterial cell, and if you offer the bacteria, oh, my bacteria, please take this recombinant DNA happily inside you. So the bacteria will say what? Bacteria will say a big no to you. They're very reluctant to intake a piece of recombinant DNA inside. Why it's so? Because the nature of DNA, you know, the nature of DNA, that is negatively charged, as well as the cell membrane, the proteins associated with cell membrane, they are also negatively charged. So obviously negative will repel negative charge. So it will not be very welcoming process, but artificially you have to do something so that the bacteria will welcome and intake the recombinant DNA inside the cytoplasm. So for that, first you have to treat the bacteria with cold calcium chloride. Now these calcium ions, they will go and bind with the probable negative points on DNA as well as on the negatively charged proteins of cell membrane. Okay, and that will also, this calcium chloride, that will make the cell wall and cell membrane proteins more welcoming for any foreign molecules. So that will create temporary channels, that will open temporary transferrin channels on the cell wall and cell membrane. And next, you need to make the bacteria more vulnerable. That's why you are applying heat shock. So what is heat shock? Once you are putting the bacteria, treating them, keeping them inside an incubator with 42 degrees Celsius, then you are transferring them into a media with ice, that is 0 degrees Celsius. So within few seconds, if you are doing so, then what will happen? The pores of the bacterial cell wall and cell membrane, they will forcefully opened, and by utilizing that opportunity, will be inserting our recombinant DNA inside the bacteria. Right? Now let's talk about plants. So process is very easy. You have to use biolistics or gene gun. Biolistics or gene gun. So basically, these are microscopic gun will coat the recombinant DNA with particles of tungsten and gold. So here what we use? Particles of gold and tungsten, as because these are comparatively inert metal and very effective to penetrate through the cell wall of plants. So you will be utilizing these particles, tungsten and gold particles, wrapped because this will be wrapping the DNA, and will be, by utilizing this particular gene gun or bialistic, will be throwing them inside the plant cell cytoplasm. Okay. So these are the processes which are generally used to transform cells if you are reluctant about the vector mediated transformation. So we are done with the topic that is tools of recombinant DNA technology, and we are here, we will talk about the process of recombinant DNA technology, the process how to produce rDNA, recombinant DNA in our laboratory in a stepwise manner. Okay, so the topics given in NCERT we have to study accordingly. First, the process of isolation of DNA, either it can be from a plant cell, animal cell, or from bacteria. Then cutting the DNA into proper pieces. Then the process of amplification of DNA. So basically we have to study the topic of PCR, polymerase chain reaction. Then how to insert the recombinant DNA into a competent host. Then after forming a recombinant organism or genetically modified organism, we have to produce the product. That's why we need to study about the obtaining the product. Here we have to use bioreactors, and after formation, after getting the recombinant proteins from them, the last topic we have to study, that is downstream processing, the process of separation and purification. So let's start with isolation of DNA. Okay, so when we are talking about isolation of DNA, we have to collect pure DNA from cell. So for that we need help of some of the important enzymes. Okay, so we can isolate DNA from a plant cell, from animal cell, or from a fungal cell, or from a bacterial cell, right? So let's study how to isolate a DNA from a cell. So obviously, what are the envelopes of a cell present inside, present outside? There is firstly there will be cell wall, and the component of cell wall is different into different organisms. Like if we talk about bacteria, it's peptidoglycan. If we talk about fungi, it's chitin. If we talk about plant, it's cellulose and hemicellulose. Then we have to, after digestion of the cell wall, we need to digest the cell membrane, and we have to purify the DNA from other impure macromolecules, especially from RNA, from proteins, and from lipids. So in stepwise manner, we'll study how to do so. Okay. So we need some enzymes, like we can use lysozyme, we can use chitinase, sometimes we have to use cellulase, we have to use enzymes like protease, we have to use enzymes like RNase. Yes. So basically, when you are when you want to digest the cell wall of bacteria, you need to apply lysozyme. That digests cell wall of bacteria. Now, what about chitinase? That digests cell wall of fungi, because that's made up of chitin. And if you want to digest the cell wall of plant, you have to use cellulase. Okay. So cellular envelope is gone. Now you can treat them with detergent for dissolving the phospholipids and other lipids present in the cell membrane or inside the cytoplasm. So when you treat that mixture with detergent, any kind of detergent, that will react with the lipid molecule and eliminate them out. Now you have impurities like protease. So how to kick out protease? How to kick out protein with the help of protease? Okay. So these are protein digesting enzyme that digests proteins. And when to use RNase? To kick out, to eliminate RNA. So all these things are acting as impurities. Now after eliminating all these things, you are gonna get pure DNA. Pure DNA is collected and isolated from test tube. So you can observe 3 test tubes. So what is the process of isolation of the DNA from this fraction? So basically, this is the first test tube what you have to put here, you have to take a paste of tissue. It can be plant tissue, it can be animal tissue. Then with that we have to add some liquid detergent with that, as well as we have to put some enzymes depending upon the tissue. If you are using plant paste, then you have to take which enzyme? You have to take cellulase enzyme, right? Now that will digest the plant cell wall, and the detergent will digest the cell membrane, and the cytoplasmic impurities are there. So now you will use protease and RNase. Now everything is digested except the DNA. Now you have to separate it out. Now how to separate out the DNA? How to identify? You have to add some chilled ethanol, right? So for the process of this purification and isolation of DNA, what you have to use? You have to use chilled ethanol, or also you can use chilled isopropyl alcohol, right? Either you can use ethyl alcohol or isopropyl alcohol. Whenever you will be adding this chilled ethanol, so automatically you can observe the fine strands of DNAs are getting separated, fine strands of DNA are getting precipitated near to the surface. Now with the help of a sterilized rod, it can be a rod of steel, it can be sterilized rod of glass, you have to take the fine strands of DNA out from the test tube, and the process we call it spooling. So the process of collecting the fine strands of DNA from the mixture with the help of a sterilized rod, the process, what do you call? The process is called as spooling. Remember this. So the question can be asked, the spooling process is used during the time of isolation of DNA. So now you have the purified DNA along with you. Now next, what you have to do? Now you have to cut the DNA in a specific manner so that the gene of interest can be obtained from that large molecule of DNA. So basically what the DNA molecule, whatever you have collected, that is a large twisted strand, that is a whole DNA, right? So next thing you have to do, you have to treat the DNA with restriction endonuclease. In shortcut, I am writing it REN. Now after treating with restriction endonuclease, you will be get some small, some moderate and some large size cut piece of DNA strand. Now next what you have to do, you have to run this DNA strand, as because you want to separate them out based upon their size, and you know the process. You have to perform gel electrophoresis. When you are done with gel electrophoresis, by the process of scooping and elution, you will separate out your target DNA strands. You will separate out your target DNA strands, and what do we call them? GOI, gene of interest. Now the next thing what you have to do, by utilizing the similar restriction endonuclease, you have to produce cut in a gene vehicle, that is you have to take one of the plasmid, and you have to insert this gene of interest with the help of enzyme called ligase. You have to paste the gene of interest, insert that gene taxi or gene vehicle or plasmid vector. So now rDNA is formed. Now the next thing what you have to do, so in laboratory you have produced very minimal quantity of rDNA. Now before performing the transformation experiment, you have to multiply, you have to amplify this rDNA into millions of copies. So the next step will be amplification of DNA. So the next step will be amplification of DNA, and for this process of amplification you need help of the procedure called polymerase chain reaction. Okay. So by utilizing this process of polymerase chain reaction, within a short span of time we can produce billions copies of this rDNA. Suppose these are the copy paste version of our produced rDNA into our lab. So let's study the process of PCR. The full form is polymerase chain reaction. Polymerase chain reaction. Now why do we perform this polymerase chain reaction? The aim of PCR is amplification of DNA. Amplification of DNA, basically what is amplification? Production of multiple copies of a sample DNA, right? And the machine which is used for this process, the machine is called thermal cycler. This is the machine which is used to undergo polymerase chain reaction. Yes. Now the process is done into 3 major steps. So let's talk about that. First step, that is called denaturation. So what will do in denaturation? Basically, if you have a single stranded DNA with you, double stranded single sample of DNA with you, a double stranded DNA as a sample with you, you have to separate the strands from, separate the complementary or anti-parallel strands from each other. So that is the process of denaturation, and this process of denaturation is like it needs high temperature for separation, for breaking down the hydrogen bonds in between the anti-parallel strands. So the temperature you need here near about 94 degrees Celsius. Okay, right? And you have to keep these strands for few seconds, like 55 to 60 seconds. Then automatically you can observe these strands are separated from each other. One strand is 5' to 3' strand, and another strand is 3' to 5' strand. That's why we are calling them complementary but anti-parallel strands. So now these strands are separated, so that's why we are calling them the process of denaturation. Okay. Now the second step will perform. Second step, that is called annealing. Now what do you mean by annealing? We have to anneal, we have to tag DNA primer. Basically they are small strand of DNA, single stranded oligo nucleotide pieces, that is called annealing. For the process of annealing, we need to maintain a little low temperature. For the process of annealing, we need a temperature of 55 degrees Celsius. Remember the temperature, what kind of temperature we are using. So for annealing, what do we need? We need oligonucleotides, and these oligonucleotides are called DNA primer. So we need to use complementary DNA primers to tag them with the DNA strands. So we have two DNA strands with us. One 5' to 3' strand, one 3' to 5' strand. Now we need to tag these oligonucleotide DNA primers towards the 3' end. Remember, where we are tagging, we are annealing the DNA primers towards the 3' end because they are complementary. They also have 5' to 3' end. Each primer has a 5' end and a 3' end, right? Each primer has a 5' end and a 3' end. So this kind of oligonucleotides strands are which are complementary to these sample DNA strands are annealed with both of them. So this is the process of annealing. Now we are going forward towards the third step, that is called elongation or extension. Now what we do, basically we try to copy the cellular mechanism of a DNA replication, that is called as the process of elongation and extension. So for extension or elongation, basically we will replicate both the strands, right? We'll produce multiple copies. So for this process we need something, we need enzyme called DNA polymerase for the process of elongation. And obviously for elongation we need supply of multiple deoxynucleotides, DNTPs. We need multiple DNTPs, deoxynucleotides, right, to tag them with this primer to produce antiparallel chains, right? So here for DNA polymerase, we need a special kind of DNA polymerase because everything, whatever we are doing, that is happening into a high temperature, that is not maintaining a 37 or body temperature, that is above that. So this process of elongation is performed at a temperature of 72 degrees Celsius. That is quite high, right? So for that we need a thermostable DNA polymerase, not a normal DNA polymerase, and that is obtained from obviously one of the archaebacteria, that is called Thermus aquaticus, which we are gonna get at hot spring or hot vents, right? So basically they are habituated with high temperature, living in high temperature. That's why their enzymes are also modified, right? So why we cannot tolerate high temperature or low temperature? Because of our enzymes. But why those archaebacterias, they have the ability to survive into extreme condition? Because they have special enzyme, and for the bacteria called Thermus aquaticus, they have a special enzyme called a special DNA polymerase called Taq polymerase. In shortcut form, you can call it Taq-Pol. Taq-Pol is the DNA polymerase obtained from a bacteria called Thermus aquaticus. Now, with the help of this DNA polymerase, what we'll be doing here, we are gonna extend the DNA strands. We are gonna extend the DNA strands, that means the anti-parallel strands. From 3' to 5' end. So already where to start? Near to the primer attached. Now this thermostable DNA polymerase, what it will do? It will utilize the DNTPs, deoxynucleotide nucleotides, and it will elongate the DNA strand. And thus, from one single strand, two different strands are produced, two new strands are produced, so the DNA is said to be amplified or multiplied. So this is about one cycle. This is one cycle. So more the numbers of cycle you will perform, the multiple copies of DNA double strands we are gonna get, right? So that was all about PCR, polymerase chain reaction. Now few more important thing I'm gonna write here. So as per NCERT, if you perform 30 cycles, so after running 30 cycles, this is one cycle, so after running 30 cycles, how many copies of DNA we are gonna get? We are gonna get near about 1 billion copies of DNA. 1 billion copies of DNA we are gonna get after running 30 cycles. So now you can ask me, sir, then sir, what will be the formula? How we can calculate after 2 cycles how many DNA we are gonna get? After 10 cycles how many DNA strands we are gonna get? After 30 cycles how many DNA you are gonna get? Right. So first you need to learn some signals, that is N0 and N. So N0 means the number of initial strand, the number of initial DNA, double stranded DNA, that we will be calling at N0. So if you are starting the process with one double stranded DNA, so N0 will be 1. If you are starting the process with 2 double stranded DNA, so N0 will be 2. If you are starting the process with 25 double stranded DNA, so N0 will be 25. And N is the number of cycle. N is the number of cycle. So the formula is like that. It's N0 into 2 to the power N, and we are gonna get the number of copies of DNA. Number of copies of DNA. So if you got a question that someone is performing polymerase chain reaction into the lab and he started with one single copy of double stranded DNA and he is running 20 cycles. So after completion of 20 cycles, how many copies of double stranded DNA he or she gonna get? So how you will be calculating in that case? You will be calculating, he started with one double stranded copies of DNA and he is running how many cycles? He is running 20 cycles. So it will be 2 to the power 20. So whatever the amount you are gonna get, that will be your answer. So that's a homework for you. You can please calculate accordingly and comment into the given comment box, right? So I think the process of polymerase chain reaction is understood, we know. So whenever in our daily life we need to copy something, we can produce multiple copies with the help of the Xerox machine. So in case of biotechnology, the Xerox machine for DNA is thermal cycler, and the process is called polymerase chain reaction. Okay. Remember, it has 3 steps: denaturation, separation of DNA strands. DNA has 2 antiparallel chains, so you have to separate out them with the help of heat. Now next is annealing. Now your aim is to produce multiple copies. You have to imitate the process of cellular DNA replication. So for that, cell needs RNA primer. Here we are attaching DNA primer, and then with the help of special DNA polymerase, that is called thermostable polymerase, or one of the example is TAC-Pol, which is obtained from Thermus aquaticus, and with the help of multiple deoxynucleotides or DNTPs, we will be elongating the antiparallel chains of each parental strands, and we are going to get multiple copies of DNA, right? Now, when you are done with PCR, what is the next step you have to perform? You know the next step. After performing the PCR, you got multiple copies of the recombinant DNA. Now you have to make the cells or the host cell competent. You already know the process of making the host cell competent. Either you can, if it's a bacteria or prokaryote, you can use calcium chloride and the heat shock method, or if it's a plant cell, you can use agrobacterium mediated transformation by utilizing that TI plasmid. If it's a dicot, if it's a monocot, directly you can inject the recombinant piece of DNA with the help of gene gun or ballistics. If it's an animal cell, directly you can inject with the help of micro injection, or if you want a vector mediated transformation of animal cell, then you can use disarmed pathogens like disarmed retrovirus. Okay. Now after that, when you are injecting the recombinant DNA inside the host cell, that cell is transformed. Now the next step, you have to culture, you have to produce multiple copies of your host cell in your conditioned laboratory environment. So here you will be using now bioreactor. So the next topic we are going to study, we are done with isolation of DNA, we are done with cutting of DNA, we are done with amplification of DNA. We already know the process of insertion of recombinant DNA, right? Next, we are left with obtaining the product and downstream processing. So let's start with the process of obtaining the product. Obtaining the product. So what is product for us? Product will be recombinant proteins. Or enzymes or hormones or antibodies or antibiotics. So we call them recombinant proteins. So for these we need bioreactors. We need tissue culture facilities. Now when we are talking about tissue culture facilities, we need some container. It can be a simple flask, it can be bioreactors. Now inside those containers we have to pour the nutritious media that are gonna provide nutrition to the culturing cells. Either they can be bacterial cell, or they can be plant cell, or they can be animal cell. Now whenever you are culturing bacteria, you need different set of facilities. When you are culturing plant cell, you need different set of facilities. And obviously when you are culturing animal cell, this quite complex method. You need sterilized and well managed lab facilities or bioreactors. So basically, this kind of machines are used for large scale production. We call them bioreactors. Bioreactors are used for large scale production of recombinant proteins or our desired product. Okay. So when you are talking about tissue culture facilities, so there can be different types. This can be continuous culture. This can be batch culture. This can be a fed batch culture. So one by one we'll study this. For culture, you can use simple flasks. For culture, you can use bioreactors. And the question can be asked that what is the advantage of bioreactors over flask? So in course of our study, we learn obviously. So for large scale production, bioreactors are helpful for us. In that case, we can't use flask as because the environment inside the containers of bioreactors, they are highly regulated. You can regulate pH, you can regulate moisture, you can regulate temperature. Each and everything can be regulated. But for flask, once you have poured the culture media, there is no chance of regulation. So in case of flask culture, obviously you have to follow the batch culture method. But in case of bioreactor, you can follow either continuous culture, either batch culture or fed batch culture. Now the question can be asked that, sir, which is more cost effective and efficient to obtain our product, either it's continuous culture or batch culture or fed batch culture. Okay, so we'll try to understand with the form of graph. For continuous culture, the graph will be like this. But for batch culture and fed batch culture, the graph is like this. So you want multiplication of your genetically modified cells or host cells or transformed host cell. For that, obviously, if you observe the graph, the log phase of growth can be seen in both the cases, but in case of continuous culture, there will be no lag phase, right? But in case of batch culture or fed batch culture, after log phase, lag phase can be or declining phase can be observed here. So obviously we prefer continuous culture, and for continuous culture always bioreactors are best because inside flask either you can perform batch culture or you can perform fed batch culture. Now what's the difference between continuous culture and batch culture or fed batch culture? Let's study that. Okay, so here I am putting the culture media. This is the total volume of culture media. The maximum volume of culture media can be kept in the container, and inside I am culturing for my genetically modified organism or the transformed cells, right? So already from the beginning we are filling the total capacity of the container in which we are gonna culture the tissue, culture the cells. So that is called batch culture. That is called batch culture. So once we are prepared, we are done with one single culture, again we have to take them out from this container. We have to put a new batch of medium and new batch of cells inside that for our culturing. So obviously you can observe, when the nutritive media is limited, the volume is limited, after a certain growth, after a certain growth of the cells, obviously there will be a saturation and there will be decline phase. So that's about batch culture. What is fed batch culture? We'll put similar number of cells into the container, but we'll put half of the volume of the culture media or the nutritive medium. Now, along with the growth of these cells, we'll keep feeding or keep adding new culture media, and it will keep on going until unless we are reaching till the maximum volume of this container. And this process is almost similar to the batch culture, but it's a little bit more efficient from the batch culture because the production will be higher in case of this fed batch culture. Okay. Now the most efficient one, that is the continuous culture. Now what happens in case of continuous culture? A certain volume of the container is maintained all the time. We'll put our culture cells or cultured cells for their growth. Continuously we'll be adding the media from one side, and after a certain time we'll be draining out the media. So what you are doing, continuously we are feeding and continuously we are draining out the excess sludge or the spoilt or the foul materials produced along with time. So in this case we can observe the highest level of growth without observing any declining curve into the cells, number of cells. So obviously this kind of facility is only possible if you are going with bioreactors. So in our NCERT, two types of bioreactors are given, right? So one of them we call them simple straight tank bioreactor, and the advanced one that is called spudged straight tank bioreactor. Now what is difference between simple straight tank and spudged straight tank? So stirring means, if this is the bioreactor and the water inside, that is the culture media, and continuously for the even distribution of oxygen if I am culturing aerobic organisms inside that. So for even distribution of the nutrients and the oxygen or the gases, we are continuously stirring it. So that's why we are naming it stirred tank. We are naming it stirred tank. Now, what do you mean by spurge stirred tank? Now, along with stirring it, we are producing source. We are applying source of oxygenation, like you have observed in aquariums or fish tanks, right? Bubbles are coming out to dissolve more and more amount of oxygen into this culture liquid, into this like nutritive liquid inside it, that we call culture medium. So obviously whenever we are producing or supplying direct air or oxygen into the tank along with the stirring process, it gonna be so much more efficient in compared to simple stirred tank. And obviously you need to follow the proper diagrams given in NCERT along with their leveling. So for moving the stirring plates like flat blade like structure, we need to provide them force from motor. What about this particular measurement clock? We can measure the pressure of the gas produced inside the tank. You need acid base or pH control system. We need steam sterilization mechanism inside it before putting the culture media inside that bioreactors. We need to sterilize the tank because we only want the desired microbe or desired cells to grow inside it. We don't want unwanted growth of cells or contamination. Okay, at the surface we need foam breaker because fermentation is going inside. Sometime fermentation will take place inside. So obviously upon fermentation there will be formation of froth or foams. Now we need to add special blades along with the main axis of the propeller or the motor as foam breaker. Okay, and obviously the medium which are using, that we are calling as culture broth. Culture broth is nothing but the culture media. Culture media which has species specific or cell specific nutrition. Different bacteria or different, if you are culturing plants, if you are culturing aerobic bacteria, if you are culturing anaerobic bacteria, so different organisms need different requirements of nutrients, right? So that was all about the bioreactors. Now what after bioreactors? Downstream processing. Remember this thing, D equals to S plus P, DSP. So downstream processing means D for downstream processing. S for separation, and P for purification. So once your product is prepared inside the bioreactors, you need to separate out and purify the product. You need to collect the product, and the product has multiple impurities. Now with lab test we will eliminate the impurities, that is called purification. And upon purification you will store it and you will send into the market with proper leveling. So that is the whole process starting from collection of or selection of DNA, collection and isolation of DNA, formation of recombinant one, transforming the cells, forming genetically modified cells, genetically modified organism, and then culturing them inside the special facilities or bioreactors to produce your desired product. So, we are done with this chapter. We will solve few questions. Okay. So, one question is there, statement based question again. So, statement 1 is saying the first restriction endonuclease, that is Hind2, whose function depended upon a specific DNA nucleotide sequence, was isolated and characterized 5 years later. Absolutely true statement. Statement 2 is saying that Hind1 was isolated before Hind2. Yes, we know the nomenclature of restriction enzyme, right? So, the Roman numericals we use, right, the Roman numbers, that is the order of isolation. So, Hind1 was isolated before Hind2, but the function or the discovery of this enzyme as a efficient restriction enzyme was first for Hind2. So, both the statements are true here. So, both the statements are true. Let's solve the next question, assertion and reason question. Okay. So, the assertion is saying in the year 1963, two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. So, in the year of 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. That is true fact. One is methyltransferase and one is restriction endonuclease. The reason is saying that because both of them were restriction endonuclease, this is false. So, it is very easy to answer, assertion is true, but reason is false. Another assertion and reason. So, the assertion is saying that this stickiness of the ends facilitates the action of enzyme DNA ligase. The stickiness of the ends facilitates, if I am talking about restriction endonuclease, upon their sticky end cut, obviously the stickiness will facilitate the action of enzyme DNA ligase. That is a true fact. Now, let us see what reason is saying. The reason is saying they form hydrogen bonds with their complementary cut counterparts. Okay, the sticky ends will be forming hydrogen bonds with their complementary counterpart, but when you are talking about DNA ligase, it will form the it will rejoin the cut piece of sugar phosphate backbone. So, basically that will create if I am forming sticky end cut, obviously the sticky end cuts will rapidly produce hydrogen bonds with them, and then ligase will come to form the to paste the sugar phosphate backbone by forming the phosphodiester bond. Yes. So, the reason is again true, and we can say that yes, the reason is justifying the assertion. Now, let us talk about this question. assertion. There are overhanging stretches called sticky ends on each strands of DNA upon the action of restriction endonuclease. That is true. The overhanging stretches means that sticky ends. Reason said that restriction enzymes cut the strands of DNA little away from the center of palindrome site but between the same two bases on the opposite strand. Exactly. If you are talking about echo R1, the palindrome site is follows GAATTC from 5' to 3' end, and for the complementary strand again it will follow GAATTC. So that is called palindrome. GAATTC, that is called palindromic sequence. Now what is the center? Center is this, center of palindrome. This is called center of palindrome, COP. Now our echo R1 will not cut at the center, it will produce cut in between, little away from the center, in between G and A on 5' to 3' strand, and similarly to the antiparallel strand it will produce cut little away from the center of palindrome in between the nitrogenous base called G and A, guanine and adenine. So in both the cases is forming cut in between G and A, and later on it will produce sticky end. So reason is also true, but is it justifying, satisfying? There are overhanging stretches called sticky ends because restriction enzyme. Yes, reason is justifying assertion. Now another question. Restriction endonucleases are enzymes that process pre-RNAs? No. Are enzymes that splice DNA? Exactly. Restriction endonuclease protect bacterial cells from antibiotics? No. None of the above. So we got our answer. Restriction endonuclease are enzymes that splice DNA molecules. Okay. Next question. A gene is said to be cloned if the DNA sequence of the gene is known. Not the answer. The function of the gene is known. It's not the answer. There is a DNA probe for the gene. This is also not the answer. If the gene has been isolated and copied. Exactly. You have taken out a gene of interest from your source organism. Now you will put this gene of interest into the host. Now upon successful replication of this host inside the host, when the host is producing the offspring, inside them also you can get the stress of this gene of interest and their product. So that's what we call, it's called isolation and copying. So when the gene is able to copy or inherit its copy into the offspring upcoming generation of the host organism, then it is said that the gene is cloned. So we got our answer. So with this I am completing the chapter, the first chapter of biotechnology unit, that is biotechnology principle and processes. I am also coming with other chapters from class 11th and class 12 with similar one short lecture with detailed explanation but following the NCERT. Yes, till then it's bye bye, thank you and Jai Hind.